Yeah, thank you very much for coming here, and it's a great pleasure for me to give a talk about sequence analysis on a HPC conference. It's the first time for me. I will tell you a bit about SeqAn, which is a library which I develop in my lab since about 10 years. And it's a generic library, it's a template library. And I will tell you a little bit about the content and how we address modernizing the code. But before I do that, I start basically with an intro. Why do you need SeqAn, right? And that is a thing which I think not many people appreciate if you're not in the field. Can I maybe ask, there are like, what, 10 people, who of you actually ever worked with sequencing data, genomic data? 1, 2, 3, 4, well, about half. So for the others, what are we dealing with? This picture basically is very nice in showing that. It just shows you a double helix of a genome. And what is more, it shows you little reads, little pieces of the genome. And from the very beginning in the mid-90s, this hasn't changed. I will come to advances in biotechnology later. But the fact is that although we are dealing with genomes which are, ours is about 3 gigabases big and others are even bigger, all we can read are little reads of length 200. And what you can do with these reads, that actually has changed quite a bit in the last years. Here's one of my favorite quotes from '97. That's a paper in genome research, which is a high impact paper in our field in bioinformatics. And that was written by Phil Green in answer to a paper written by Gene Myers, who was at the time at University of Arizona. And he said, well, if I have these little reads, I can actually assemble a whole genome, which later led actually to the assembly of the Celera genome with Greg Venter. And he wrote a rebuttal and saying, here I quote, however, it's clear upon reflection that unmapped genomic reads are an extremely inefficient way to obtain biological information and are virtually useless for most purposes. So that was in '97. And just to counter that a little bit, I got now this quote. It's from the McKinsey Global Institute in their report about disruptive technologies. And that report contains advances that will transform life, business, and the global economy. So according to McKinsey, the economic impact of next-generation sequencing, just of sequencing, they estimate that it has a potential economic impact of about $700 billion to $1.6 trillion per year by 2025. So I think they're not virtually useless. Because they will affect lots of areas, many areas of our life. It will be agriculture, right? Monsanto and all these guys, they're sequencing plants and they're trying to increase the yield of food plants. Metagenomics, right? We will not only look at our own genome, but also our gut genome, right? There's like many, many bacteria and viruses inside us, and it's clear that you can derive a lot of medical information from analyzing the metagenome. That means all the genomes that are, for example, in your stomach. Cancer. It's now actually quite standard that cancer patients are sequenced during the progression of cancer, and one tries to find out what happens during the progression. Or we will, of course, be able to sequence now whole pedigrees and elucidate the causes for hereditary diseases much better. So there are many applications for an NGS. And why has that exploded so much? And that is probably little appreciated. I mean, you always see this bioinformatics and sequencing, but this has changed. It was a giant leap in biotechnology. This is a picture from Celera, and you see basically the sequencing facility of Celera, and on the right you see a guy standing there. So it's a huge factory hall, and one of the technicians is standing at a sequencer with an attached computer. And this sequencing facility, which was at the time the biggest in the world, was running 24/7, and it could sequence about 100 million base pairs per day. Right? So what has changed is basically this. This factory hall can now be replaced by one machine. That's an Illumina HiSeq. And that Illumina HiSeq can sequence now 100 million, 100 billion base pairs per day. So if you do the math, it means that within roughly 10 years, sequencing has become about 10 million times cheaper. And I don't know any other part of business which has seen such a decrease in cost. So what is now actually possible is we can sequence everything, really everything. If you do the math, I found this paper, number of species on Earth tagged at 8.7 million. So if you assume that each of these species has a genome of about 10 to the 8 base pairs, that means we can say the Earth's genome has about 10 to the 15 base pairs. So I talked to some people at Illumina, and they say worldwide there's a population of about 1,000 of their sequencers now around, which is of course expanding all the time. Each of the HiSeqs can sequence 10 to the 11 base pairs per day. That means the Earth genome at 10x coverage could be sequenced within 10 days. So we can sequence everything we can get a hold of. We can sequence humans not once, but several times. And it will cost basically nothing. It will be a standard routine diagnosis if you go to the doctor, if you have just some indication. So that really changed the game. We have now a technology which allows us to acquire 10 to the 14, 15 base pairs. And people are doing that, right? Iceland has sequenced the whole population. Saudi Arabia started a project. England will do a 100,000 genome project. So many, many more people will get sequenced. And so we have to deal with analyzing this data. And here software libraries come in. This is like how I see the world in bioinformatics. It's basically divided into two extremes. On the one side, there is a more experimental-oriented bioinformaticians, and they basically care about their analysis pipelines. They want to analyze RNA-seq data, chip-seq data. They want to find structural variants in cancer, and so on. And they usually have some programming skills or not. They're interested in analysis pipelines, maybe maintainable tools, but then it pretty much ends. On the other side, we have the computer scientists. They are experts in data structures, multi-core, secondary memory, fast I/O, and so on. And they are mostly interested in theoretical considerations, how their data structure works, what is the runtime, in algorithm design, and then maybe prototype implementation. So they're not so much interested in robust systems that work, which might actually be a bit different in the HPC community. And in the middle, I think it's very nice if you have a software library, right, where you can include all the good things from the computer science side and then offer it to people with less expertise in that field. And that's why we started the SeqAn project. It took 2.5 years, and we had like basically 6-7 design phases of ideas how we would achieve our goals. And then 2008 we had the first publication coming out where we had a first set of data structures and algorithms. Meanwhile, just the SeqAn library is cited more over 360 times, and if you take all the citations of tools that use SeqAn, you have many, many more. And I had a student once take out the institutions that use SeqAn, and I omitted the German institution. So you see it's Harvard Medical School, it's the EMBL, the Craig Venter Institute, Princeton, Yale, IBM Watson Research, etc. So it's actually quite nice that we have now a large user base. Since a couple of years, we put SeqAn under the BSD license, so it's now free for academic and for commercial use. And since then, we're actually also collaborating with a couple of companies. So that was my introduction, and now I come to more what is SeqAn, how is SeqAn designed, and then about I will tell you about the content in SeqAn and what we want to do within our Intel HPC. So what are the three basic principles of the SeqAn library design? It is basically three things, and you see it here in some actual code in red here. The first is that we use template subclassing. So to introduce polymorphisms, we do not use classes, but we use templates. And you see here as a type def an index, which is a string index where you can do fast searches. You have a template argument for the alphabet type. You have a template argument for the implementation. The second you see here is global functions. So you have a global function interface to work on the data structures. And the third we have is meta functions, so we can derive type information at compile time. Right? So these are the three basic design principles which actually took 5-6 iterations trying out different ideas. Now I come to the SeqAn content. What is in there? So if you do NGS analysis, I can't give you a complete overview, but I try to pinpoint the highlights in my opinion, and also the parts where we will start with a generic parallelization and modernization of the code. So first of all, which is very often used, is we support all the important file formats for high throughput sequencing analysis. You see it here, FastA, FastQ. Basically on the right you always see actual SeqAn code that would work, right? You have a sequence stream or you have a BEM stream, and you can read zip BEM files and or write them. So for read mapping we support SEM, BEM, hopefully soon also CRAM. We have a very nice unified full text indexing framework. So the interface is for all the implementations the same. So we have for suffix trees, if you think suffix trees, we have the implementation of suffix arrays, enhanced suffix arrays, lazy suffix trees. We have FM indices and different implementations of QCRAM indices. And again, the interface, you see the code, it's very easy. You say index of a type of sequence, that could be DNA, it could be proteins or some other alphabet, and you specialize it with an implementation template argument. And all these indices support multiple strings and external memory construction and usage. You basically have just another template argument saying external, and then the index lives on your disk if it's too big. All the indices support a sequential finder interface, and obviously we will also move to an asynchronous parallel finder interface, but you can in that little code piece here you see how you would find in an index a string, right? And you could iterate over all the positions in your index text in your genome. All the suffix trees, the indices, can be accessed with iterators that support different traversals, top-down, depth-first search, random, etc. And we also have some predefined specialized iterators, like for iterating over all repeats, over all supermaximal repeats or maximal unique matches in a collection of strings. The parallel interface supports exact and approximate string matching, so you can search for strings with errors in the indices. And we have different QCRAM filters for counting and seed filters. In the index interface, upcoming is that we will have a new interface for bidirectional indexed search with a bidirectional FM index, which supports then searches in O of 1 or O of log sigma time per character. So now I come a little bit into more detail, which is our pairwise alignment module, because here I can show you later that we take an advantage of our generic design of the library. So pairwise alignment is easy enough. What you want to do is you want to compare two strings, and you see on the right a dynamic programming table. And in this dynamic programming table, you compute, given some score function, the best score of an alignment. So you would have an initialization phase, 0, and then you would fill the first row, and you would fill the first column. Having done that, you can now go row by row or column by column, and you have this recursion formula which takes a maximum of 3 values. And if you look at the first free cell here, where there's now the 0, it's a maximum of the 3 cells in the diagonal on the top and on the left, right? And then you would just fill this matrix. And then depending on the version of the alignment algorithm you do, you can compute a traceback. That means you look where you got the maximum from. And if you trace back this maximum path, it corresponds then to this alignment which you see below. So you would align T with a T with a T, then you would have a gap with an A, G with G, and T with C. So far, so good. And there's many libraries and implementations, I will come to that later, which do global alignment. This was an example of global alignment. There's also local alignment, and there's a gazillion of sub-variations of this. And the nice thing in SeqAn is that we support them pretty much all in an interface which can be configured at compile time. So for all your dynamic programming-based alignment algorithm, it could be semi-global alignments, local alignments, split breakpoint. If you don't know them, it doesn't matter. The point is we have about 198 different versions of alignment algorithms which can be configured at compile time. How do we do that? Well, we wrote the code such that here you see two matrices. One is an unbanded alignment and the other is a banded alignment where you only compute the cells for a striped version of this dynamic programming matrix. And you see different colors. And basically at compile time, our algorithm basically colors the cells where the green color, for example, says you only have to go to the left or up, and the blue color means you only go diagonally or up. So basically, we know then for a certain cell what to do with it in the algorithm. And they're all combinations within this tagging system. And by configuring then the algorithm, we know exactly what to do with one of the cells, right? So this is all done at compile time, and that gives you then efficient code for over 200 versions of pairwise alignment. And the thing is, we only have to know now how to compute for one of these colored cells the basic operation. Like in the little example, it was a maximum of 3 values. That can be configured with some template arguments. Here just some examples. Semi-global alignment, linear gap costs, no traceback. You would write that in SeqAn like that. You would configure it. You can have local alignment, affine gap cost, single traces. You can align the gaps left, and so on and so on. This might, for normal bioinformaticians, look a little bit daunting. So we also wrap that and have standard interfaces for global alignment, for local alignment. And we will have wrapper interfaces, not only one sequence against one, but many against many. And I will come back to that when I'll come to the parallelization. So how fast is this generic library-based alignment? It's actually quite nice and fast. So in a one-to-one interface, we just have here 2 sequences. This here is an affine alignment of 10 kilobases of a dengue virus. You see the comparison in runtime with other publicly available implementations. And only the NCBI C++ implementation of the global alignment is a little bit better. But that implementation only supports global alignment. So we have basically 200 types of alignment which can be configured. So keep that in mind. We are also in the one thread case already quite competitive. So what else do we have? Where is SeqAn strong? We also support compressed data structures. So for example, in SeqAn we have a data type string set, which not surprisingly is a set of strings. But the nice thing is you can specialize it and say, I have a journal string set. So what does it do? It basically only stores one reference string and then only the differences. So we do a referential compression. If you have, for example, here the source and the target, then we don't store all the sequence which is already in the reference. You see the two, the C and G in blue and green. This is data which is not in the reference, so we need here an insertion buffer. So we store a pointer to the reference. We have an insertion buffer for sequence that is not in the reference. And then we have a binary tree which allows us to do logarithmic time searches and indexing of this reference string, and also a linear time reading out of the whole string. In such a tree, we have so-called journal entries, and these 4 numbers are basically the virtual position in the string, the physical position in the reference or in the insertion buffer, and then the length of the sequence, and finally just a bit which tells you whether the sequence is in the insertion buffer or in the reference. So having that, we can easily access every position in the string at a little runtime penalty. So once we have this data type, what we can do now, if we have now a string set, right, we can basically encode all the strings very efficiently in a so-called delta map, write that to disk, and having just the reference and this delta map, we can easily reconstruct the whole string set. So far, so good. You could say, well, okay, now you can have a reference-based compression of a few strings. That's not so exciting. But what we did with that is we have now a data type which lets you iterate, and we call that data parallel, over a set of strings much, much faster. So what is the task? Imagine you have a couple of strings, for example, 1,000 genomes which are from individuals which are very much alike but have some differences. And you have some algorithm which scans linearly over the sequences, right? So it needs a sequence context, it computes something, and it has maybe an output. So you usually would, of course, write a loop and you would scan over all these sequences, right? Linearly. Obviously, you can make an OpenMP loop about that, but we'll come to that later. The point being, if these sequences are very much alike, I mean, you do a lot of work in vain. So what we do now in a data type which we call journal string tree is we process multiple similar sequences at only once. If you look at this example in dark blue, I depicted now the sequence which is unique, and in light blue is sequence which is just repeated. And what we do now is, if the algorithm now scans over the sequence, it goes over a sequence context, and it always asks for the next character, and it does some computation. And then it comes to such a branch point, and the next character is either here or here. And this data type just enumerates then all possibilities. That means it will put your sequence down here, your sequence context. And if you now find something, for example, you can ask the data structure, where am I? And it will tell you, for this example, you're in sequence 1 and 3 at the following positions. And then it would move the context up here. If you run over here and you find something, you can ask the data structure, where am I? And it would, at this case, now say you're in sequence 0, 1, 2, 3 at different positions. So it does all the bookkeeping for you. And so on, right? Basically, this gives you a list of sequence contexts, and it guarantees you not to omit any of the sequence context. So what does it bring you? That gives you already a speed up in data parallelism, and we haven't done any SIMD acceleration or any multi-threading or acceleration here. So if you take our data type and just take it for one sequence, so there's no benefit in taking this data type, we have a little penalty of 15%. So you see here the number 0.85 is a runtime factor compared to the trivial algorithm. But if you go to 129 sequences, and these are sequences of chromosome 1, chromosome 1 is about 200 million base pairs. So we have here 129 times 200 million base pairs. And if you run trivially over these sequences, it takes you 719 seconds, and with our data type, only 13 seconds. So only double the time it takes you to run once over chromosome 1. And if you go to 1,000 sequences, the factor is about 80 times which we are faster with a single core, just by data parallelism. And the other nice thing is, if you look at the memory, you see on the very right column that we stopped at 26 gigabyte, where we just put some sequences out and we reloaded the rest of the sequence. So it's a total worth of 50 gigabyte of sequences we look at. And on the second last column, you see that our top memory consumption was for 1,000 sequences 2.5 gigabyte. So it's very space efficient, and you can iterate as if you run over lots of similar sequences. that was published 2014 at HITSEQ. Okay, so I stop here now with giving you an overview about the content of SeqAn. There's much more which I can't go into, and of course the next question is, well, that's all good, and what applications actually do you do with that? And we have several. I don't go into many of my lab. I just show you some screenshots, and in red is basically always publications that use SeqAn which are not from my lab. In green is which are related with my lab. And you see that the Salzburg lab here from Johns Hopkins, they use it. Top Hat, Bowtie uses SeqAn. In green you see our publications, yeah. And this is just a screenshot I took from Google Scholar. So actually I should count them, but many tens of publications use SeqAn actively, not CIDRs, but really use it. I just wanted to point out one of our publications because which is a read mapper. It's called YARA, which stands for yet another read mapper because there are so many publications in that field. But the nice thing is this is now a culmination of, I think, 8 years of work in read mapping, and I'm really happy with this read mapper now. So the YARA can work on Illumina HiSeq, MySeq, single end, paired end, made pairs on INE, torrent, proton, PGM. Has a very low memory footprint. It is multi-threaded. It maps over 10 gigabase pairs per hour on a laptop. It uses compressed I/O, which is very fast. Very few parameters only. It can be exact. There's no more heuristics in edit distance alignment, so there's no heuristics in this algorithm. And we have just a collaboration with Molecular Health in Heidelberg, and they're evaluating it independently, so we actually don't know how, but they compare it to NovoAlign, and we hear it's comparable. NovoAlign is a commercial aligner which is very accurate, but quite slow. Here you see basically the precision of YARA. In the left, the big numbers, 100%, are just an average of these small numbers. And in the first row, you see with YARA 100, 100, 100, 100, 99.8, 99.4. These are basically, the first cell means these are all the reads with 0 errors. How many of those did you find? And then come all the reads with 1 error, with 2 errors, with 3, 4, and 5 errors. So what you see is that YARA and GEM, which has a very similar algorithm, are very, quite accurate. And at the moment, leading software, Bowtie 2 and BWA, are actually not. So they're not finding all the positions even with 0 errors because they have heuristic algorithms in them. And I said we can be completely accurate, but you see that for 4 and 5 errors we have here not 100%, and that is basically because we do in the seeding phase only a Hamming distance alignment and not a edit distance alignment. We can do that, so we could change that at the cost of a factor of 2 in runtime. So we decided at default, we leave it at Hamming distance, and we only lose very, very few alignment with high errors. Right? So, but the algorithm has no heuristics. We could just switch it to edit distance. And you see in the throughput, we have 13.5 gigabases per hour, which is about 3-4 times faster than BWA. And it uses just the standard FM index in memory. Okay, what are we going to do with the SeqAn IPCC, which we actually start 1st of December. So I'm a little bit ahead of time here, but we actually went to 2 rounds of applications, and in the meanwhile, we actually could already do some work, even without the support of Intel, but that will become now even better. What we thought is, we have some applications like YARA are of course multi-threaded, but we did it on the application level. And in our proposal, we think it makes much more sense to do it on the library level. So we want to basically modernize the whole SeqAn library using multi-threading and vectorization. So we want to introduce an abstraction layer for SIMD vectorization, hiding the different intrinsics you have depending on the hardware system. We want to introduce an abstraction layer for multi-threading, either with OpenMP or with the TBB. And obviously, for us, the Xeon Phi is a very nice hardware architecture for the kind of computations we want to do. So we want to address both Xeon and Xeon Phi. And then we want to use SeqAn's generic design to accelerate the core algorithms wherever possible by multiplying SIMD and multithreading in the library and not on the application level. And the first thing we did here is basically the alignment example. So remember, I showed you that our one-threaded standard DP algorithm is already quite competitive, quite fast if you compare it to other implementations. And what we did is we have now also a many against many interface. That's not new. This here is a picture taken from a publication of Rocknes from 2011. He reviewed some of the efforts done so far for SIMD parallelization for dynamic programming-based alignment. You see Wozniak, the standard approach is going via the anti-diagonals because then you don't have any dependencies. So given the black anti-diagonal, if you have that, then you can compute the blue anti-diagonal in one step. There are other versions of parallelizing many sequences against one. But what we chose is actually what Rocknes did in the figure D. So we really take many against many sequences, really multiple data and single instruction, right? Because this maximum computation of one cell, and you look at the surrounding cells, is always the same. And remember that we configured and we put the colors on all these cells using template programming. And that doesn't change. We don't care whether it's SIMD or not. So basically, we only had to replace a couple of basic functions in the SeqAn design. So instead of having now this DP matrix on two sequences, we run it, and we have a template argument on sequences of SIMD vectors. Right? In this toy example, you see 3 sequences vertical and 3 horizontal, and it's always vertical 1 versus horizontal 1, vertical 2 versus horizontal 2. There's no cross product. It's 1 against 1, 2 against 2, and so on. That means, we could basically, the whole logic stayed the same, and we had to just replace a couple of functions which, instead of taking a maximum depending on the scoring scheme of 3 cells, we replaced this function with SIMD instructions. And that worked, first of all, very well. And for all these 200 combinations of DP matrices, not only one, but for all. Here you see the speedups. In green and in red, it's DNA, which has simpler scoring schemes. The maximal speedup on this machine was 16. It was AVX on an Intel i3, actually. It was on the laptop of the student. And we get speedups near 16 already for very short sequences. And the other 2 curves are for proteins. For proteins, you need a lookup in a protein scoring matrix, which is not easily to parallelize. And the other people actually did some pre-computations and tried to vectorize that. And the students basically said, yeah, I don't have time to do that now in my master season. He didn't, and he did a serial lookup, and it worked perfectly well. So we got a speed up of 8 to 10, where the other implementation didn't. I'll show you just some results. So we were able now to SIMD accelerate the whole alignment module. And if we combine that now with multithreading, how would it fare? So I don't do now runtime. I actually returned that now in the many-to-many interface. If you compute 2 million pairwise alignments and you take our already nice, fast, one-threaded code, we got a speed of about 2,700 alignments per second. If you do it on 16 threads, 30,000 alignments per second. If you do it on one thread with SIMD, you get 54,000, and all these bars are actually to scale. If you combine it, we can compute 2 million alignments in 3 seconds, full DP alignments. So that worked actually now very, very well, and you see the power of combining the SIMD and the multi-core implementation. In the second experiment, what we did local alignment, where there is a dedicated library by Zhao and Mars and colleagues. It's called SSW. so SIMD, Smith-Waterman library. It does exactly, again, one type of alignment. And we just configured our module to do local alignment. And what you see on the left, you see what we did is we took different lengths DNA and scanned it via a database and aligned it with a database of DNA sequences. On the left, the 3 columns, you see that for DNA we are very comparable. For the shorter we are a bit slower. For the longer we are a bit faster. And on the right, you see that what I told you, the simple strategy for the proteins works very well. So we are 3 to 4 times faster than the SSW. And we have a module which works basically for all kinds of alignments that you want to do. So what's next? As I said, we will start in December with a new student working on this. And we are also in touch with some Intel engineers. It's Pascal Constanza and Johnny Hancock from the UK. And we provided them now with a framework to go from this many against many alignment interface to the one against one, right? Because we don't have that yet. And for large sequences, that might be very nice to have. And the first idea is what we want to do is, you'd see here these 3. First of all, we go via the anti-diagonals. That's not new. And you see these 4 green blocks. So that's not one cell, but we will compute now larger blocked alignments. So one thread will now take basically 16 of those and use our SIMD accelerated version of the many against many interface. And the second thread will take the next 16 and so on. How to schedule that and in what order we want to compute it, that is now the question we want to address with the Intel engineers. The second thing in the IPCC is what we want to do is adding to this data parallelism, which I showed you in the journal string tree, and multiply this speed up we have already there, which is about a factor of 100, with multi-threading and SIMD. So that's potentially, I think, we can have here speedups of several orders of magnitudes. Because if you think about it, what we do there is we effectively construct a list of consecutive sequence contexts. So it would be very easy to compute block boundaries that can be generated on the fly using one thread, or pre-computed. And then basically we can have a pool of sequence context which can then be given to several threads or some algorithm who in turn will be accelerated using vectorization. Yeah, that's pretty much it. You can follow us. We also have a Twitter account, SeqAnLib, @SeqAnLib. You see the website, www.seqan.de. And if you're interested, we have yearly user meetings. We are also supported in the German network for bioinformatics infrastructure for this library. In March 30th to April 1st, there will be a user meeting. The date is already fixed, and on the website, there will be probably in 1-2 weeks, there will be a program. And we will tweet it then around. So thank you.